CoPheScan methodology and a few results with CoPheScan using simulated and real data. So coming to phenome-wide association studies, the phenome-wide association studies, they study the association between a single variant and multiple phenotypes. This is an inverse of the PheWAS approach, which studies a single phenotype versus multiple variants. So the phenotypes in the phenome might include electronic health record data, lab measurements, transcriptomic data, and other omics data, as well as medical imaging data. So PheWAS helps in exploiting the genetic pleiotropy. So it helps in drug repurposing. So if you have known associated drug targets, you test for other associations and thus helping in repurposing. Additionally, if you have known associations and you test for multiple associations, and if they have a direction of effect opposite to the disease, these can be potential adverse effects which can be monitored in the trials. Next, coming to some of the PheWAS challenges. So the main challenge that we are addressing is LD confounding. So this is PheWAS Manhattan plot. You have the phenotypes on the x-axis and the p-values on the y-axis. And what happens in PheWAS is you identify potential phenotypes, but you do not know if these phenotypes, the variant is associated with these phenotypes, or it might just be an LD and you just it just shows up here. So this is one of the things we want to address. So what are the current PheWAS approaches to to combat the LD confounding? So what is done is there is a two-stage approach. PheWAS is performed first, and following that, they estimate the posterior probability of the variant being causal for a pair of traits. So this is the basis of the widely used CoLock method. So this was done also by Veturi and colleagues, where they developed a unified framework to study links between plasma lipids and diseases. So this framework included GWAS, TWAS, PheWAS, and Mendelian randomization. Within the PheWAS part, they used CoLock to remove LD contamination. So now coming to the new methodology that we have developed is called CoPheScan, or CoLock adapted phenome-wide scans. So this reformulates the CoLock hypotheses. In the CoLock, we test if two traits have a shared causal association. In this case of CoPheScan, we have a causal variant which is already known to be associated with the trait, and we test for causal associations with multiple traits. So on the left, we have the traditional PheWAS with p-values and identification of potential phenotypes. With CoPheScan, we can simultaneously identify associations as well as find out if the variant is an LD confounder or not. So coming to the CoPheScan methodology, as I said before, so we already know the causal variant which is associated with the primary trait, and we want to test for associations with the query trait. CoPheScan discriminates between three hypotheses. So HN, where there is no association with the causal variant, and HA, where the association with the variant other than the causal variant. This can occur in n-1 ways, and then there is HC, which is association with the causal variant. And this occurs only in one way because this is the variant of interest. So the inner workings of CoPheScan are such. So you have the input of CoPheScan is multi-trait GWAS summary statistics. And the priors and base factors can be calculated in different ways. So you have the fixed priors, which are derived from CoLock, or hierarchical priors. So I'll be explaining this further in subsequent slides. So you also have the base factors, which are obtained in different ways. One is the approximate base factors method, or using SUSY. So once you have the priors and base factors, you combine them to obtain the posterior probabilities of these three hypotheses. And so you have a phenome-wide posterior probability of causal association of the variant with your phenotype. So coming to the calculations of the prior. So as I mentioned, you have PN, PN, PC. So you have the prior probability that no SNP is associated with the query trait, and PA that it is associated with an alternate variant, and PC where the causal SNP is associated with the query trait. So we derive these. When I say fixed priors, it means that the priors are obtained from the default prior set in CoLock. And the hypotheses priors from these are same for the HN and HC, and HA we multiply PA into n-1. If you have a large enough data set, you can also infer the priors using a hierarchical Bayesian model. So the likelihood here is given by this term equation, and the terms BFAK and BFCK are the base factors at alternate variants and the base factors at the causal variant. The priors can be given as such, so they sum up to 1. And we introduce these hyperparameters as alpha, beta, and when we have covariate information, we also include another hyperparameter, that is gamma. In our experiments, we include the genetic correlation, so we have included that here. So we sample these hyperparameters using a Metropolis Hastings algorithm. So the base factors can be calculated in 2 ways. So you have under the single variant assumption, you have the approximate base factors, but we also know that within a given region, you might have multiple variants. So to address that, we have also incorporated SUSY, which is sum of single effects. What SUSY does, it returns credible sets, which each contains highly correlated SNPs. And you have multiple base factor vectors, and we run CoPheScan on each of these. So once you have that, so either of these base factors can be plugged in here, and finally you get the posterior probability of the 3 hypotheses. So coming to the results with CoPheScan. So we used simulated data as well as real data. From the simulated data, we simulated 10,000 case control GWAS summary statistics. So different ratios of HN, HA, and HC traits were simulated. Next, in the real data, the phenotypes in the phenome-wide scan were from the UK Biobank of the non-cancer illness code, which is a self-reported. So these were 283 traits. Then coming to the variants, we obtained these from 2 diverse sources. One was from the FinGen disease endpoints, and another one was from PQTL data from Workingstaff et al. And et al. So the FinGen, what we did was we used the FindMap data from FinGen as well as the PQTL data. And the putative causal variants were selected as such as when they were present in a credible set size of 1. Once we had the variant and phenotype combinations, we ran all the CoPheScan comparisons as possible. So when we come to the simulated data, our model was able to discriminate well between all the true HN, HA, and HC scenarios. As we see here, we have a high posterior probability of HA for all the true HA cases, and a high posterior probability of HC for all the true HC cases. So as I mentioned before, we detected the putative causal variants from 2 different sources. So from the FinGen disease endpoints, we were able to identify 135 variants, and we saw that 33 out of 283 traits were associated with at least 1 variant. So you had these matched traits between the UK Biobank and FinGen, the ones which we used for identifying the variants. So the ones which were matched, out of the ones which were matched, 27% were seen to be causally associated with a posterior probability of greater than 0.5, while in the unmatched traits, it was 0.2%. Similarly, with the PQTL variants, in the data, we identified 1,887 variants, out of which 60 out of 283 traits were associated with at least 1 variant. And here we identified 0.02% of all the phenotype variant combinations. So what we see is when you're using 2 different sources, you might have a different proportion of traits which are identified. So this might lend itself to actually using a hierarchical model. Okay, so this is a figure where we have used a hierarchical model to infer the priors. And on the X and Y axis, you have the posterior probability using hierarchical priors and without using the genetic correlation, and also by including the genetic correlation data. What we see are some phenotypes actually have a higher posterior probability of causal association when we include the genetic correlation. So in case of diabetes and pernicious anemia for these 2 variants. Okay, so next few slides, I will show you a few known associations we identified as well as some novel associations. So we just to explain these plots, you have the PheWAS p-value plots, which you traditionally get with the current PheWAS methods, and these are the CoPheScan results. So on the so the nodes on the top, in the top box are the FinGen primary traits from which the variants were identified. Then you have the variant, and then these are the UK Biobank query traits which were identified using CoPheScan. So we put the threshold of 0.5, and we were able to obtain the known associations of PTPN22 with autoimmune diseases. Yep. Next, we also identified known associations with SERPINA1. SERPINA1 is known to cause alpha-1 antitrypsin deficiency, particularly this variant we identified, and also associated, in turn associated with emphysema, COPD, etc. So the FinGen primary traits from which the variant was identified and the UK Biobank query traits are similar. Next, we look at the PQTL dataset and UK Biobank phenotype combination of results. So you have this PCSK9 protein and the RS11 variant, which was identified. And as expected, it is associated with angina, high cholesterol, and heart attack and myocardial infarction. So PCSK9, you have currently PCSK9 inhibitors to lower LDL cholesterol. Next, we identified novel association with the POMGNT2 protein. The POMGNT2 protein is involved in the glycosylation of the alpha-dystroglycan, and which is required for maintaining the stability of the muscle cell membranes. And so some variants of POMGNT2 are known to be associated with muscular dystrophy. In our analysis, we were able to identify some novel associations with the cranial nerve palsy, pneumothorax, vertigo, and hypopituitarism. So this is something we'll be investigating further. So finally, CoPheScan is an approach to PheWAS. It allows for the investigation of pleiotropy. So when we see a causal association with CoPheScan, we can say that it is indeed causal and it's just not in LD with another variant. And it's fast, and we can, given a large enough data set, we can also infer the priors, and we're also able to exploit covariate information. As we saw with our genetic correlation results, we actually identify more phenotypes. So I would like to thank everyone who was involved in the project and our collaborators. Thank you.
Very nice job. Are there questions for Ichcha? Yes, microphone. Thanks.
Very nice, thank you. Sorry if I missed this. So you fine map the genetic variants to hone in on variants after accounting for LD. When a variant lights up for multiple traits, are the trait associations adjusted for one another? I sort of missed that.
Sorry, I didn't get that.
Are sort of when you say have 4 traits linked to one variant, yes, are there, say, a bunch of other correlated traits that you've filtered away due to correlation?
No, there is no filtration. So all the traits are included, irrespective of the correlation. Yeah.
Cool. Thank you.
Any more questions? Well, I'll ask you, when you design your simulation studies, so what was the logic behind that, and which, I mean, you show a couple of the scenarios, and why you chose those?
So, we simulated in different ratios because we know that HN cases are far more than the HK and HC cases, and for the results that we showed, the HA cases have an alternate variant in the trait, but the causal variant is not present, and in HC, the causal variant is present. The HA and HC are much lower than HN, and we wanted to see if all of them are identified. Next, we'll see if there are multiple causal variants, we're able to identify those as well.
Thank you.
Okay, so we have a question on Zoom from Chiara Auwerx. Would you like to unmute yourself and say your question?
Sure, I can. So I was wondering if you could use well-powered molecular QTL studies to identify causal variants and then perform CoPheScan using those. And then kind of, I was maybe struggling a bit to understand how would that be different from just running colocalization using between molecular QTLs and then the GWASs.
Okay, so in colocalization, you do pairwise comparisons of all of the traits and the EQTL studies. Here we're honing in on just a known causal association. So the 5 different hypotheses of Kolok just reduced to 3. So instead of performing multiple pairwise comparisons, you actually get down to
Thank you.
comparison.
Any more questions? Okay, well, thanks very much, Ichcha. We now move to Chiara, who's going to tell us about copy number variants, how affect complex traits and disease.
This one. Maybe this one.
Good afternoon, everyone. So my name is Chiara, and I'm a PhD student at the University of Lausanne. And I'm really happy to be here today to talk to you about our research on the individual and global impact of copy number variants on complex traits and common diseases. Oh, sorry. So copy number variants, or CNVs, are a type of structural variation that is usually characterized by either the deletion or the duplication of a DNA fragment of at least 50 base pairs. Now, in the field of medical genetics, CNVs are known to act as potent phenotypic modifiers, as they have been found to be the genetic etiology of several rare and debilitating genomic syndromes, as is shown here on the right. However, historically, CNVs have been quite hard to assess, and as a consequence, their role in complex traits remains poorly understood. So the way one typically investigates such complex traits in statistical genetics is through the GWAS framework, where for each of the SS probes, one simply performs a linear regression that estimates the phenotypic consequence of carrying an additional copy of the effect allele. So we propose here to adapt this framework to the study of CNVs by simply substituting the classical genotype readout for the copy number stage of CNV proxy probes. And now, of course, the first step in order to perform such a CNV GWAS is to call CNVs in our population of interest, which in this case would be the UK Biobank, that offers multiple advantages. So first of all, it is a relatively large population of about 500,000 individuals, meaning that we can have enough statistical power in order to detect associations with events that are as rare as copy number variants. Now, in a second time, individuals are well phenotyped, so we can assess the role of CNVs on a wide variety of complex traits. And finally, but very importantly, individuals who are not recruited based on clinical phenotypes, meaning that we can assess the role of CNVs in a rather healthy general population. So we then used PenCNV in order to call the CNVs, but also applied a quality score pipeline that in a first time will attribute a probabilistic quality score to each of the called CNVs. And this is reflective of the likelihood that this is a true positive. But in a second time, it will also transform the linear genomic coordinates of the CNV call into a matrix format. And this allows us to work at the probe level, and thereby we can bypass one of the common hurdles when working with CNVs, that is linked to the fact that even for recurrent CNV loci, there is variation in the boundaries and breakpoints of the CNVs. So we then stringently defined high confidence CNVs and encoded them in a plain format, which allows us to perform efficient genome-wide association studies. And now a small note regarding the samples: we restrict the analysis to unrelated individuals of white British ancestry. And we also exclude individuals with a report of blood malignancy in order to protect the analysis from the impact of somatic CNVs. Then for the probes, we require a minimum CNV frequency of 0.005%. And we also perform pruning. Oh, it's not showing anymore. Oh, okay, sorry. Yeah. But so the idea of performing pruning at this high threshold is to group the probes that are at the core region of the CNV, but still maintain some flexibility around the boundaries. And we then selected 57 different medically relevant continuous traits that we integrated into our pipeline. Now, a small note on the last step: we work with 3 different association models. So the first one would be a mirror model that is analogous to a classical additive model used for SNP GWAS. And here we simply assume that deletion duplications have an equal size effect, but in the opposite direction. And then we also work with 2 CNV type specific models where we simply disregard one type of CNV carriers, and those would be the duplication only and deletion only models. So using those different models, we then perform the GWAS and finish with a stepwise condition analysis that allows to make sure that the signals are independent. And this resulted in the identification of 131 independent association signals over 47 different traits. And those are all represented on the map that you have here, but it's rather complex. So I highlighted an example to show you how to read it. So here highlighted in yellow, we have a deletion because of the brownish color on chromosome 1q21 that is associated with decreased level of serum urate as indicated by the minus sign. At the top of the graph, there's also the duplication and deletion frequencies that are plotted, and this is an important information to keep in mind because often we detect more associations for the CMV type that is most frequent in our cohort. Now, the reassuring thing is that we were able to reproduce several known associations. So, for instance, at the top here, you would have the association between CMVs on the 16p11.2 region and BMI. But what was previously not appreciated was the extent of pleiotropy of these recurrent CMV regions. So if we take again the example of the 16p11.2 deletion, we found it to be associated with 26 different traits. Another recurrent pattern that we observed was that for about 38% of our autosomal CMV signals, we do identify a SNP-GWAS signals for the same trait within the CMV region. And I will illustrate this with a specific example, which is the association between a deletion on chromosome 1 and decreased level of serum urate. So the first thing that we did was to map the CMVs onto the chromosome, and we observed that it overlapped with a gene called PDZK1. And this gene happens to be a very well-known SNP-GWAS signal for serum urate levels. And in addition to that, we know that the gene is expressed in the kidney and that it encodes for a scaffold protein that interacts with various urate transporters. So it already was a very good candidate gene, but then we took it a step further and split the deletion carriers into two groups. So the ones that have the large deletion versus the small deletion that only affects PDZK1 and a few adjacent genes. And what you can see from the box plot is that it's sufficient to carry the small deletion in order to observe the phenotypic consequence. So that means that with this evidence, we can be very confident that PDZK1 is causing the observed association. And I think that this example illustrates well how CMVs at SNP-GWA loci can actually contribute to phenotypic variability in the general population. So coming back to the main result map, another pattern that we observed was that many of our signals mapped to regions that were previously involved with Mendelian disorders. And for these, we tend to observe variable expressivity and penetrance. And once more, I will illustrate this with a specific example, which is a deletion on chromosome 12 that we found to be associated with increased level of bilirubin. So again, when we use the approach to map the CNVs onto the chromosome, we found it to overlap with two genes in red here, SLCO1b1 and SLCO1b3, which both are known to encode for hepatic transporters involved in the uptake of bilirubin. And now, unlike the previous example, here we see that individuals with a large deletion that affects both of the genes suffer from a significantly higher increase in bilirubin, but that still remains within the healthy range that is indicated by the green background. And where I think it becomes very interesting is that there's an extremely rare autosomal recessive disorder that is called Reuter Syndrome. And this disease is genetically characterized by the homozygous mutation or deletion of both of these genes. And the main clinical manifestation of this disease is actually hyperbilirubinemia. And we see very nicely that the very few individuals in the UK Biobank that have this diagnosis, well, they have extremely high and pathogenic levels of bilirubin. So with the second example, I wanted to show you how CMVs at Mendelian disorder loci can actually impact disease-associated phenotypes in the general population, but without necessarily causing a full-blown diagnosable disorder. So up to now, I've focused on CMVs in isolations, but it's also possible to aggregate them in order to look at their global impact on the phenotype. And the first step to do that is to calculate each individual autosomal CMV burden, either as a number of megabase pairs or genes that are affected by them. And we can then use these newly calculated matrices in order to perform linear regression with the same 57 traits that we assessed with the CMV GWAS. And you can see that 35 of these traits seem to be negatively impacted by having a high CMV burden. So for instance, we observed that it leads to an increase in adiposity, a decrease in physical capacity, as well as elevated levels of several renal and hepatic biomarkers. Now, another conclusion we can make from this graph is that the deleterious impact of a high CMV burden seems to be mainly driven by the number of deleted genes. And this can be seen from the fact that we have more color in this column, which is for the gene burden, and that we also have more brown color, which is standing for the deletion burden, versus the green color, which stands for the duplication burden. So we then went a step further, and we correct for what we call modifier CNVs. So these are CNVs that by themselves affect the trait of interest, as identified with our CNV GWAS. So if we take the example of the burden for urate levels, we would correct for the CNV region that contains PDZK1. And to our surprise, 30 traits remain significantly associated with the burden, meaning that for them, there is a highly polygenic CNV architecture. So in the previous slide, I showed you that having a high CMV burden is globally negative for your health. But we then ask ourselves whether the negative impact of the burden also extended to some more general aspects of an individual's life. And indeed, we found that having a high CMV burden had a rather negative impact on several socioeconomic factors, as well as on longevity, which we assess with two proxies, parental lifespan or age, which we use as a proxy for survivorship. So overall, this shows that the deleterious impact of a high CMV burden is likely to ultimately result in a decrease in longevity. So all the results I've presented so far have been published in this month's edition of the American Journal of Human Genetics. So if you're interested in more specific examples, I invite you to have a look. But to conclude the first part of my presentation, I would like to leave you with a few take-home messages. So the first one would be that we developed a CNV calling and GWAS pipeline that we then use to show that CNVs are pleiotropic phenotype modulators that play an important role in shaping complex traits in the healthy population. We also use this data to show that having a high CMV burden had a globally negative consequence on an individual's health, and it also led to reduction in longevity. And finally, the polygenic CMV architecture we observed for so many traits suggests that in the future, once we will have larger cohorts with more statistical power, we will be able to detect new CMV trait associations. So for the second part, I will now switch to unpublished data. And I think that a rather natural extension of the work I presented so far would be to interrogate whether CNVs also affect susceptibility to common diseases. And to do that, we will simply switch from using a linear regression to using a logistic regression that allows us to investigate how CNVs impact disease probability. And in the context of this project, we also work with a fourth association model, which is a U-shaped model that has a rather intuitive assumption that deletion and duplication affect the phenotype in a similar manner. So we then manually created a set of 60 diseases based on ICD-10 codes and also generated 2 associated burden matrices. And as you can see, we really tried to select disorder that spread the different chapters of the ICD-10 code, but we still try to favor disease for which we suspect high heritability and for which we have a decent number of cases. And as a proof of concept for this method, I will now show you an association we found for ovarian cancer. And this association mapped to the BRCA1 gene, which is encoding for a tumor suppressor gene that is known to be one of the leading risk factors for developing ovarian cancer, but also breast cancer. And if we now highlight the individuals in red that have the diagnosis, we see that we have 2 out of 12 deletion carriers that do have a diagnosis of ovarian cancer, and this proportion is much higher than what we have for copy-neutral individuals. So this association is a bit to be expected because BRCA1 is a known risk factor, but it still shows that our method is able to detect biologically meaningful associations. So I will then switch to a second example. This time it involves chronic kidney disease or CKD. And so in the past, previous people have shown that the deletion of the RCAD locus, which is this one here, as well as point mutations in the mediator gene HNF1B, highlighted in red, are causing renal cyst and diabetes, which is a relatively severe disorder that is characterized mainly by structural and functional defects of the kidney, as well as maturity onset diabetes of the young. And so with our continuous trait CMV GWAS, we were able to show that the duplication of this region as well is causing alteration in various renal biomarkers. So if we now zoom in on a specific trait, such as, for instance, cystatin C, we do see that indeed the dosage of the region seems to have a U-shaped effect. And this is very well corroborated by our U-shaped model for ICD-10 CMV-GWAS that finds that alteration in the copy number of this region leads to an increased risk of developing CKD, as is shown by the fact that we have 7 affected individuals among our 121 CMV carriers. So with this final example, I just wanted to show you how our method is able to detect disease associations at regions that we previously found to alter disease-associated biomarkers. So to conclude this second part, I showed you that the ICD-10 CMV-GWAS is able to recapitulate known associations, and that we also do observe co-localization of CMV signals for diseases with CMV signals for disease-related biomarkers. And I will conclude with a small word of caution, because as a reminder, we work with CMVs that have a relatively low frequency, and we also have an imbalance in the case control proportion, meaning that we're really at the limit of what we can detect, and we need to be extremely careful when validating the associations that we find. And with that, I would like to thank everyone who's been involved in this project, especially people from the Kutalik and Raymond group, as well as our Estonian and Swiss collaborators, of course, the biobank participants, and finally the EMGM committee for giving me the chance to present our work. And with that, I'm happy to answer questions.
Nice talk. Any questions for Chiara? Yes. Okay.
So I've got one quick one and then one wacky one. So the quick one is, have you looked at the exome data and tried to get CMVs from that in biobank?
So no, because it took us a time to get access to the exomes. I think we just got it like maybe last week. So we haven't, but we know of people that had them before us, and we're thinking also to collaborate with them to validate some things. But I would like to point out that, so we use the genotype data, so we have the large CMVs. I think with the exomes, you will have the small ones. The ideal is the genomes, because then you have everything. So
yeah, coming soon. But the wacky question is, if you take your CMV burden and consider it as a quantitative trait and run a GWAS, do you find anything?
So if we do, we haven't tested that. So you mean that we no?
I'm
okay. So we haven't tested exactly that, but I can tell you about something we tested that goes a bit in the direction. So, but it's not exactly the same, no. So maybe I can still tell you. So, you know, for the ICD-10 codes, we also have the burdens, but this is like more the disease burden. And the disease burden is associated with some regions. And those happen to be like the recurrent CMV regions, like the 16p11.2, that is also highly pleiotropic for the continuous traits. But we haven't done the burden GWAS, no.
I can't decide whether it's interesting or not. It's sort of, are there genetic variants which affect your propensity to have CMVs? So I don't know.
Yeah, I mean, it's possible, because
thank you. It was a really nice talk. Where you have the CMVs co-localizing with a known SNP signal, is that because the SNP is in LD with them, or are they two independent effects on the same gene?
this is
Is the SNP like an EQTL for the gene that the CMV deletes?
So it's a really good question, because this is something we will explore in the future to see if, like, for instance, the CMV is tagging, because it's more likely that the CMV is causal than a simple SNP, because it's so also many of the CMVs we talk about, they're large and they also affect multiple genes. So one like tricky question will also be to identify within those maybe 3 or 4 genes that are affected, or maybe even 10, which one is like causing the phenotype, or maybe it's a combination of multiple ones. So far we've only looked at like if you take the genomic coordinates, are they located at the same place? But we've not done further analysis. But this is one plan for the future. So
thank you.
I hope that soon will be able to answer that.
Okay, so we have a question on Zoom from Stefan. Stefan, can you unmute yourself and say your question?
Can you hear me?
Yes, we can.
Yeah, my question would be about the p-values you calculated. They are based on very few events. Can you trust your asymptotic approximations.
So the p-values we calculate. So you mean for the IC10 GWAS?
Yeah, I think that the last one where you mentioned you had just 7 events.
Yeah. So I know this is a very big limitation. So this is just doing the GWAS. So one step that we're working on, but I haven't got the data yet, is to implement permutation tests to see how robust the associations remain. But yeah, it's something we're not going to be able to change. We have few individuals that have CMVs plus diseases. And I think it's an intrinsic limitation of the data. I hope this answers the question.
Yeah, thank you.
Excellent. Well, if there, okay, one last question.
First off, lovely talk, doing lots of stuff we were dreaming of doing 10 years ago. A lot of the illustrations you gave had very variable endpoints, and you were getting associations at the edges of those calls. How confident are you that you really do have the variable endpoints rather than that you have some confounding that is causing you to see the variation there only in individuals with particular conditions?
This is a good question, and we really have done no work on that to verify that. Then we often have like the signal that like really spreads, like it's not that you have only a signal at the edges, it's like also in the middle region, but it's true that often that you have like small bumps. One possibility is maybe that you also have some individuals that have shorter CMVs at those regions, because those are the classical breakpoints that tend to often have CMVs, and that maybe because you have because we work with few cases, each case counts, and if you have 1 or 2 more, maybe that leads to a stronger signal. But we have not tested if this could be due to confounders or something.
Excellent. Okay, well, thanks very much for the talk. Our next speaker is Venn, who's going to talk us about Flashfm visualization method.
Okay. Good afternoon, everyone. Thank you for coming. In my presentation, I would like to introduce Flashfm-ivis, an interactive visualization tool for fine mapping methods, particularly useful to explore multi-quantitative traits. This is a joint work with Jenny and Adam. And if you have any question, feel free to email me. And also, if you want to download the whole source code about this package, go to this GitHub. Currently, it's purely written in R because we want to encourage users in the field to use it. But we produce a web-based R-Shiny application. So if you are interested, you can just go to website and try it in the real time. You don't need any programming experience. Also, libraries we included in the package are very common in the Python community as well. So if you are good at Python, maybe we can extend the platform, the package to Python language as well. So a brief background before we demonstrate our visualization tool. So 2 years ago, in this same meeting conference, Jenny presented a work with her collaborators. They named it Flashfm, which stands flexible and shared information based in joint fine mapping. Basically, we don't have the time to explain those the detail in this section, and you can go on the GitHub, or there's a paper you can read later. And but the key message is Flashfm as an R package or a method try to combine, try to joint multiple quantitative trait analysis. And because we know related disease and traits likely share some cause variety. So if we can combine those shared information, we can get more useful maybe to discover the cause varies or something. And on the other hand, if there's no any shared covariate or shared information, Flashfm will give the similar result to a single trait phi mapping. However, by combining those multiple quantitative traits, it will create some new challenges. For example, the first part challenge is it will bring more complexity and maybe take more computational power, or we need a different kind of data set. So the original Flashfm addressed those issues. And the second part is about how to, yes, we can get the meaningful result from combine different kind of information, share the information together, but it will give us more challenge to how to understand those data and what kind of information, the way they share their information and give us meaningful result. So Flashfm-ivis is try to answer or try to visualize those stuff in order for the users to better understand the result, particularly the complex result from different combination information set and the different interactions about those informations. If you want to know the detail about the method and the series stuff, you can go read this paper published already. So my presentation will focus on the visualization part, how to understand the output from this method. So basically, we call it Flashfm-ivis because it's interactive and the users can play with it and they can install it on the RStudio and run it on the local machine. But we create a web page based RShiny so you can use it without any program language experience. It's very flexible. It tries to connect the result from different traits and combine them to understand the shared information set. For the better user experience, you can go on those RShiny website to try by yourself and play with this visualization tool. Although we call it Flashfm-ivis, we try to show the result, better understand the result from Flashfm, but we also extend the tool for a more general purpose. So users can basically, if they run any phi map methods, let's say phi map, if they follow our input requirement, it should be able to show the result from any kind of phi mapping methods. So it's not just for Flashfm. And we compare our software with other tools is available with a very similar purpose. Up to our knowledge, the first three are particularly focused on to show the phi map result, and we add some extra features. And I'm not going to go through all those in this section. If you are interested, just go to a link. I think there's a paper link in the first slides, so you can see it in more detail. But when I demonstrate the plots, you will understand what kind of extra features we added to our package software. Also, because we have a web-based RShiny application, so if some people don't want have any program experience, they just use the website and follow the input requirement to do it very easily. So maybe it's a good way to teach bioinformatics students to understand the result from the phi mapping. To demonstrate our software, we use these examples. Basically, if you want to use a software, you just input those common GWAS summary statistics. Also, we use this data set for illustration purpose. So it is a very small data, relatively small GWAS study data, and a high percentage of related individuals. And because we are talking about multiple quantity traits, so we try to combine 4 traits. Those are the information. And if you want to understand the data better, go to the paper I just showed in the beginning of the presentation. And just note, in the plot, we often mentioned MPP, which stands marginal posterior probability of SNPs causality. So basically, it is the posterior probability that the SNPs is included in the SNP model. Some researchers may use PIP to have the same meaning of this MPP. Also, we have a PP for the SNFs models. And we also have a YouTube channel to show how to understand these examples if you have a time to watch it later. So here, yeah, one picture is worth 1,000 words. So I just go through, demonstrate the visualization package. So if you use this, let's say you use the web-based package, web-based Shiny, the first page you may think you may have some tables to check your data, whether you input the right data or not. Then you will move to this dashboard. Basically, this dashboard is like show you some individual regional association plot, or if you input other GWAS data, it may show the whole Manhattan plot. So you have because we are compare information set from different traits, different methods. So you can see here we have an individual trace, trait 1. I think people behind may not see it clearly. So you have an individual tabs to show each trait, and you have the upper panel to show the result from the single trait file mapping, and the bottom you have the multiple trait file. We use Flashfm in this example, but if some people use other file mapping method, maybe you can show here as well. So I play this video. Let's see what kind of interactive feature user can use and can play. So basically, compared to traditional regional association plot, we add some extra feature. Looks like you can, I think most of you are familiar with this plot. So you can see the size of the SNFs represent the MPP values. Also, the color represents the SNF groups. So some SNFs play very similar role in the information set. They have a same, we allocate them or group them into the same group. And if you want a particular SNFs information, you can just click it. It will show a lot of information. Basically, it's a GWAS summary statistics. So if we play this video, you can see we in this page we also included the LD matrix, LD plot, and a lot of stuff you can play with. So there's a group and the group SNFs number in each group. So it's a good way for users to check your input is right or your result is meaningful in the first stage. And the SNFs play different role or provide different information in the different traits. So you can compare them, and you can download all the plots, and you can zoom in, zoom out, etc. So you can do a lot of stuff. Also, you have some control badges, so you can define your MPP value to show those key SNFs you want to investigate further, and you can show credible sets or not, and combine different credible sets from different methods. Then we move on to the because we are talking about shared information, so we want to understand those combination of different information sets. So this is a dashboard to combine all the individual regional associate plot together. And the main feature is they are linked together. So each point on this plot are linked in different plots. So as you can see here, the left column is the result from single tree file mapping, and the right is the result from the multiple tree file mapping. And as I mentioned in the beginning, multiple tree mapping provides, if the data have shared information, they provide more accuracy or more precise result. So for our example, you can see if we only use file map single tree so the rows are the different traits. We use 4 traits in the data. So you can see the first row is one trait, another trait. So in the last trait, you can see based on the single trait file mapping, we get these SNPs, they favor these SNPs. But if we combine shared information among different traits, we get a different SNPs. And because this data set in the beginning is relatively small, if we increase the data set and combine larger Africa study, then both single and multiple show the same result. So this means shared information between traits give a similar improvement, like increasing the size of the single train analysis. So shared information is really important, and how to understand that, we can play with these interactive plots. So in here, I just show how to play it. So in the dashboard, you can see, in the web page or dashboard, you can see you have selections. If you input 2 traits, you can show only 2 traits. So there's only 2 rows. If in our case, we use 4 traits. So here we can show 4 traits, and all the plots are connected together. This means if you select the SNPs you are interested in one plot, it will link to other traits, other plots. So it's easy for you to compare different information. So you can download the plot as well. And this feature allows you to only look at the SNP group you are particularly interested and see the difference between the traits and the methods. And you can move the one plot to select particular group of SNPs and highlight them. Once you highlight them, all the plot will update automatically. So narrow down the search. And if you want to know the detailed information about these SNPs, those they will compare automatically. So you have those information at the same time. So in some SNPs play a very similar role in different traits, different methods. So you can not see a huge difference. But in some case, like the one we just mentioned in the previous slides, you can discover new information. You can make some new conclusion. For this, you can see some traits including many individual SNPs, but others may not have. Then we move to network. Because we know shared information provides very useful information, and we want to know how they connected and what kind of information they came, where those information came from. So network analysis is very useful to understand those. In here, you can see those are group SNPs. The color represent let's say you look at those SNP groups as the information you can help you to discover the cause variant, and you want to know those information where came from. So the color represent the traits they came from. So if we have this color, which means maybe the information came from those 3 traits, combine them together, and the size is about the frequencies they appear in different kind of combinations. So if the size is big, which means some SNPs in this group may interact a lot of different SNP groups. So maybe it's interesting to look at further. So we have this kind of dynamic network as well. So you can see we not only look at the SNP group, we also have individual SNPs. So if we run the network for the individual SNPs, it will have a very large network. It's very complex because the SNPs in those traits, they have a lot of individually shared information. And the color, the size, all linked. But we want to make simple, so we use SNP groups, which means SNPs in one group, in the same group, have the same information. They may play very similar roles, but because they have a higher autocorrelation or ALD, so we need to understand better about individual SNPs in SNP groups rather than only look at the SNP groups. So we move to the next plot. The next plot is try to link individual SNPs with the SNP groups. So in the network, you can see we look at separately. We look at the relationship between the SNP groups, and we also look at the relationship between the individual SNPs. Then we try to link individual SNPs with the group. So we have SNP groups created by the Flashfm, the multi-trait method, and the SNP group created from the single trait method, file mapping method. And if we show that, you can also play with this as well to understand the details. So you can see some SNPs belong to, based on the single trait mapping, they belong to a particular group, but they are not belong to any group in the, based on the multiple trait file mapping. This means maybe in the single trait, they're missing some information. They misallocate these SNPs into some groups. So you can play with this, and the size of the thickness is the MPP value, and which means the probability these SNPs is meaningful or not is belong to is the row, if the SNPs row in this group. So you can see those SNPs belong to this group and their MPP are different. So this plot is like further understanding the connection between individual SNPs and the group. Then we have the traditional Venn diagrams and the Reuter plot. Yeah, and this one is try to compare different credible set in different methods, and you can get the individual SNPs from the table. So, yeah. So you can compare the numbers in the credible set in different methods. Also, based on the Venn diagrams, you can get the detailed SNPs information, which one is belong to credible set based on different methods. And you can download the table behind to get the detailed information about SNPs. Yeah, so it's not only for the Flashfm package, it can use by other file mapping methods. So this one is just show you can input the output from the file map file map methods to show it, and you can play it on the website directly. And that's it. So if you want to learn what the series about that, go to this reference. Also, finally, we would like to thank Jenna and Ace for the suggestions. Is there any question?
Nice, Michael. We only have time for a quick question. Anyone? Well, otherwise we would just play online with it.
Okay, yeah, thank you.
Excellent. Okay, well, next speaker is Oliver, who's going to talk us about how to use genetic coloration to improve polygenic scores.
Good. Yep. Thank you. Hello, everyone. First, I'd like to thank the organisers for letting me speak today. My name is Oliver Payne. I'm a postdoctoral fellow at King's College London, and today I'm presenting some work that was done alongside Catherine Lewis, investigating how we can use local genetic correlations to improve polygenic score prediction across traits. So briefly, polygenic scoring is a way of summarising an individual's genetic risk for a given outcome, typically calculated as the GWAS effect size weighted sum of risk variance carried by the individual. They have a range of applications for research, but also personalised medicine. However, further methodological advances are needed to maximise the variance explained by polygenic scores. So, one consideration is research has shown that there is widespread genetic correlation across traits and diseases, and it's been previously shown that you can leverage GWAS summary statistics for genetically correlated phenotypes to improve prediction of a given target phenotype. And several methods exist to do this. One was published by Krappel et al. Using an elastic net model to model polygenic scores for GWAS with a genetic correlation with the target phenotype, and that showed improved prediction. And then there's other methods such as SMT-PRED, genomic SEM, and MTAG, which combine GWAS summary statistics for correlated phenotypes and then calculate polygenic scores from those multivariate summary statistics. But the limitation of these methods is that they all assume a consistent genetic correlation between the target and secondary phenotypes across the genome. However, newer methods have shown that a genome-wide genetic correlation is actually the average genetic correlation across loci. And these newer methods, such as LAVA, as well as others, can now estimate the genetic correlation within specific loci, termed the local genetic correlation, and they can also estimate the local SNP heritability. And these methods briefly work by splitting the genome into broadly LD-independent blocks, and then estimating the heritability or genetic correlation with another trait within each of those blocks. LAVA is one method to do this, recently published, but there's other methods as well, such as HES and Supernova. But they've all consistently shown that the relationship between two traits can vary across loci. So there may be, in fact, some loci that share genetic effects, whereas others do not. And there's even instances where you have a mixture of positive and negative local genetic correlations, which would otherwise not be detected by a genome-wide genetic correlation, which kind of looks across all loci at the same time. So this figure just kind of shows a description of this scenario, and you can see that if you're trying to, using multivariate methods that don't consider this, this variation in genetic correlation across loci could be a limitation. So our study aims to develop an approach for incorporating these local estimates of SNP heritability and genetic correlation to improve cross-trait polygenic score prediction. So this is summarized in this figure. In box A, we first ran LAVA to estimate local SNP heritability and local genetic correlations between our target phenotype and a range of secondary phenotypes using GWAS summary statistics. We then calculated polygenic scores using MegaPRS, which is implemented in the software LDAC, to process those GWAS summary statistics to account for LD and also perform shrinkage to improve the predictive utility of those GWAS summary statistics. And then in box B, I show that we take the score files for these secondary polygenic scores and subset the variance according to whether or not they had significant local SNP heritability for both traits or significant local genetic correlation between the two traits. And then we further reweighted the genetic effects within those loci to account for the local genetic correlation. And we tried another model where we accounted for local genetic correlation, but also accounted for differences in local SNP heritability between the traits. And then finally in box C, we modeled these different polygenic scores, the target phenotype polygenic scores, the kind of standard secondary phenotype polygenic scores, and then third, this new adjusted secondary polygenic scores that have been adjusted to account for local genetic correlation and heritability. This is a table showing a list of the GWAS we used in this study. We had 3 GWAS, each corresponding to the target phenotypes of interest, which were major depression, intelligence, and BMI. Those target phenotypes were used in the UK Biobank sample, in the European subset of UK Biobank. And then we had a range of secondary GWAS that had prior evidence of a genetic correlation with at least one of these target phenotypes. So on to some results. This shows estimates of SNP heritability and genetic correlation from LD score regression and compares them to estimates of local genetic correlation from LAVA. So I want to highlight kind of 2 scenarios. So looking in this in the top box highlighted in red, maybe I can use the mouse as well. I'll show you the pointer. So we can see here, this is the genetic correlation estimated by LD score regression between obesity and body mass index, and there is a strong genetic correlation of approximately 1. And that's consistent with the results from LAVA, which shows it found, so sorry, I haven't represented this. The LAVA results, the text inside the box shows the number of positive local genetic correlations and then the number of significant negative local genetic correlations. And we can see that in the case of obesity and BMI, the results are concordant between the two methods, with LAVA showing a large number of local genetic correlations all in the positive direction. But then there's another scenario where, which I'm highlighting here, between intelligence and depression, where LD score regression found no genome-wide correlation, but LAVA found a large number of significant local genetic correlations, which consisted of a mixture of positive and negative genetic correlations. So this kind of shows the insight that these local genetic correlation methods can provide, and we can hopefully incorporate this information when using polygenic scores for these secondary phenotypes to improve prediction. So this shows the correlation between polygenic scores for a range of secondary phenotypes predicting intelligence in UK Biobank as an example, with the on the y-axis showing the correlation between the polygenic score and the phenotype. And I'm just going to highlight two scenarios. So here we're looking at the depression polygenic score predicting intelligence in UK Biobank, and consistent with what we saw on the previous slide. So the first bar, the red bar, is just standard polygenic scores for depression predicting intelligence. The green bars are unweighted polygenic scores that are restricted to loci that have a significant heritability for both traits. And then the blue and purple bars are those that have been reweighted according to local genetic correlation estimates. And the purple bar is adjusting for local genetic correlation and SNP heritability. And what we're seeing is that when you adjust for local genetic correlation and also for differences in SNP heritability, there's a jump in the predictive utility of the depression polygenic score for intelligence, which is concordant with our results from LAVA, which showed there was a mixture of local genetic correlations that when you look genome-wide, you don't see that. But then there's the other scenario where this is showing an educational attainment polygenic score predicting intelligence, and by subsetting the loci used and reweighting doesn't improve prediction. In fact, it gets a lot worse. And this is because, probably because there isn't a mixture of local positive and negative local genetic correlations in this case. And by subsetting loci to those that could have a statistically significant heritability is removing loci that are relevant to predicting intelligence but are no longer included due to insufficient power. So then this slide shows the gain in prediction of our target phenotypes when including these reweighted polygenic scores in addition to standard genome-wide polygenic scores for the secondary phenotypes alone. So the color coding is the same as on the figure before, and what we see for all phenotypes is that there is a statistically significant improvement in prediction of our target phenotypes when including polygenic scores that have been reweighted for local genetic correlation and differences in local SNP heritability as well. And so this kind of supports the idea that, again, that reweighting these genetic effects to account for local genetic correlation can enhance the predictive utility of polygenic scores across traits. The pattern of improvements does vary across traits, and that will be dependent on whether there are secondary phenotypes with mixtures of local positive and negative local genetic correlations, and also the extent to which there are well-powered secondary phenotypes available for prediction in the first place. So then the final test we wanted to look at was, okay, well, can these reweighted polygenic scores improve prediction over and above the polygenic score for the target phenotype, which is obviously the ideal scenario where we're using all the information available. And we found a mixture of results. So first I'll talk about BMI, where we saw there was a statistically significant improvement in prediction of BMI when including these reweighted polygenic scores. So So now, sorry, the red bars indicate a model including the target polygenic scores and also the polygenic scores for the secondary phenotypes, but just genome-wide standard ones. And then the green, blue, and purple bars are including those reweighted polygenic scores. And for BMI, we see there is an improvement, but that improvement comes from just merely restricting these secondary polygenic scores to loci that have a statistically significant local heritability for both traits. For intelligence, there was a small but statistically significant improvement prediction for intelligence, and that only became statistically significant when accounting for local genetic correlations. But then for depression, the improvement actually became, well, it was roughly the same, possibly slightly worse, and this is likely due to the low variance explained of many of the polygenic scores included in the model, and it looks like it's led to some kind of overfitting. So including all of these polygenic scores has reduced the predictive utility, the prediction of depression in this case. But nonetheless, I think that when comparing these polygenic scores that have been reweighted by local genetic correlation estimates to standard secondary trait polygenic scores, we saw there was a significant improvement, which suggests by incorporating this local genetic information, it can improve the utility of secondary polygenic scores for predicting a given target phenotype. So if you'd like more information on this, check out our website called GenoPred, which includes the results and code for this study, as well as several other studies relating to polygenic scoring methodology. And this is now on bioRxiv, the results I've shown you today, if you want to see more information on that specifically. Lastly, I'd like to thank Catherine Lewis for amazing supervision throughout this project, as well as our funders and your attention today.
Thanks very much. Nice talk. Yes.
Hi, I have two quick questions. So you mentioned that you use like 2,500 LD blocks. So that would be obviously specific to one ancestry because the LD blocks are different. So that's one of my questions.
Yeah.
Yes.
So the question was that we use 2,500 LD blocks, and surely this would depend on what ancestry you're looking in, and that is correct. And the 2,500 LD blocks are those pre-specified by LAVA for a European ancestry.
So my second question is, you mentioned MTAG. So do you have any benchmarks against MTAG? Like how does your method compare?
Compares to what, sorry?
MTAG.
MTAG. So we haven't done a comparison with MTAG yet, but I would like to see how it compares. We compared it to the initial method by Krappel et al., which used the elastic net model because it was the most kind of comparable method. But I think the multivariate GWAS based methods, if they can somehow incorporate these local correlations, that could be useful. I know Genomic SCM has apparently recently included that function.
Thanks.
So Zoltan on Zoom has his hand raised. Zoltan, would you like to unmute and ask your question?
Yes, sure, thanks. So my question was, could you extend this method in two ways? One is by including multiple traits, not just one other secondary trait to help improving your project score. And second, could you combine global and local genetic correlations to make improvements? Thanks.
So I heard the first one, and in this we're using multiple secondary phenotypes to improve prediction of one target phenotype. So I think we're already doing that first one. The second part, I missed one.
Sorry, that was to combine local and global genetic correlation. Because you see sometimes global working better than local.
Yeah, so this model, the elastic net model, will already account for global genetic correlations as it uses the genome-wide kind of standard polygenic scores for those secondary phenotypes. So I think what we're doing will hopefully already kind of harness both types of genetic correlation. But thank you.
Okay. Any more questions? Okay, well, let's thanks again our speakers.